Good afternoon again. I welcome you all for my third and final session of bone tissue engineering. So, I'll just explain in a diagrammatic representation. Say this is the producer cell. This produces signaling molecules. These are signaling molecules which are nothing but growth factors. Okay, and this one is the target cell. So, we need to elicit biological response in this target cell. So, this target cell has the binding receptors on its surface. Okay. So, these are the binding receptors on the surface of the target cell. These signaling molecules go and bind to these receptors. These are the signaling molecules are nothing but the growth factors, and they go and bind to these receptors, and then it regulates the signal transduction pathways. Pathway, thereby what it do is it regulates transcription of gene present in the nucleus. Okay. Say this is the nucleus and DNA, it undergo transcription. This is the activation of gene. So, this enhances the biological response. This is how the growth factors act. So, signaling molecules go and bind to the receptor present on the target cell, then they regulate the signal transduction pathway and they regulate the gene transcription, then they elucidate the biological response. So, this producer cell has changed this response of the target cell with the help of growth factor. This is how it works. And so, in general, they are the secreted proteins that exert their effects by interacting with specific receptors on the cell surface, as I said, and through paracrine. Paracrine is nothing but where they act on the neighboring cells. The signal transfer takes place in the neighboring cells. In autocrine, as it defines, like it acts on itself in the same cell. Endocrine, where it is transferred in the blood and tissues, and it transported through the blood, and in the targeted site it will enhance its property and or elucidate its response. So, this is all about the growth factors, signaling or the signaling molecules. And these are this table shows the certain growth factors which are responsible in bone tissue engineering application. So, BMP2, which is bone morphogenetic protein 2 and bone morphogenetic protein 7 are FDA approved growth factors, and they are used in commercially available products where they help in the differentiation and migration of osteoblast cells. Osteoblast cells helps in the formation of bone, responsible for the formation of bone, and HGF, which is hepatocyte growth factor, where its function is proliferation, migration and differentiation of mesenchymal stem cells, which are very important cells. This mesenchymal stem cells, it can develop into n number of lineages, not only the bone, it can also develop into cartilage, tendon or marrow, any kind of lineages it can develop. And IGF, which is nothing but insulin like growth factor, it cell proliferation and inhibition of cell apoptosis. Apoptosis is nothing but cell death, it inhibits that IGF growth factor. And PDGF, which is platelet derived growth factor, which are responsible for embryonic development, proliferation, migration and growth of endothelial cells. So, endothelial cells are important for very much important for vascularization, which helps in the formation of new tissue. And TGF beta is nothing but transforming growth factor, helps in the proliferation and differentiation of bone forming cells, anti-proliferative factors for epithelial cells, and VEGF, vascular endothelial growth factor, migration, proliferation and survival of endothelial cells, again it is for vascularization technique. So, these are the several, there are several growth factors. I just picked up the few important growth factors in terms of bone tissue engineering application. So, there are two distinct strategies for biomaterial presentation of growth factors. It can be chemically immobilized into the substrate like polymeric substrate or the matrix, or physically encapsulation. Physical encapsulation can be done. So, there are two mechanisms on which growth factors are encapsulated into the matrix. The first one is chemical immobilization, where it involves chemical binding or affinity or interaction between the growth factor containing polymer substrate and a cell or a tissue, and this will produce the strong localized and interaction in that area. However, it is mainly based on the physical and chemical properties of the substrate as well as the growth factor dose. And the physical encapsulation of growth factors in the delivery system, it is achieved by encapsulation, diffusion and programmed release of growth factor from the substrate into the surrounding tissue. And for this, a very good example where which we saw in the last session is video where can you guess where the physical encapsulation or the physical stimulus of growth factor is done? In which mechanism? Yes, ultrasound. So, through external stimuli we are delivering the growth factors. So, this is all about the growth factor strategies and the what are growth factors and the two main strategies for growth factors. And so, to combine everything, all the scaffold, cells and growth factors, and development of an ideal bone graft substitute. So, this picture clearly depicts the properties of the ideal bone graft substitutes. First, the first one, there is the fractured bone. There is a defect in the bone. So, first thought we need to think what kind of base material we need to select, either natural material, it can be natural or synthetic polymer material, but it should be mechanically stable, angiogenic, and it should support the cell growth should be supported by the substitute material. So, the initial thinking should be about the matrix, which is nothing but the scaffold. So, scaffold can be natural or synthetic, but it should be non-toxic, mechanically stable and ideally interconnected porosity. Then the osteoconduction. So, osteoconduction is nothing but the formation of new bone on the surface. Then the recruitment of MSC cells, mesenchymal stem cells, which then differentiated into precursor cells and then into osteoblasts. So, that undergoes with osteoinduction. Recruitment of mesenchymal stem cells differentiates into precursor cells and then osteoblasts. Then the osteogenesis takes place. Osteogenesis is nothing but the formation of new tissue, which can be done with the combination of growth factors, which are nothing but bone morphogenetic proteins, where it regulate, recruits, differentiate and regenerate mesenchymal stem cells. So, osteogenesis takes place and this bone is healed completely. So, any process, it should have, any ideal bone graft should have three major processes. Osseointegration first. First, it should be osseointegrative. So, it should have all the four properties: osseointegration, osseoconduction, osseoinduction and osteogenesis. These all four important process is needed to develop a bone graft to enhance and repair the bone defect without any side effect, and it can heal completely, and it also accelerates the wound healing. So, the next few slides I will be talking about commercially available bone graft substitutes. The first one is osteograft, which is made up of ceramic material, and these are FDA approved bone graft substitutes. Osteograft, which is the name or the commercial product name, and it is made up of ceramic, and major properties are osteoconductive, limited osteoinductive when mixed with bone marrow. It has to be mixed with bone marrow in order to become an osteoinductive material cells. Bone marrow has lot of mesenchymal stem cells, and they are used for bone void filler. The next product is Nova Bone, where it is bioactive glass. Bioactive glass is nothing but the silicates. So, it is osteoconductive, limited osteoinductive when again mixed with bone marrow, and filling surgical and traumatic bone gaps. And the third one is osteosat, where it is surgical grade calcium phosphate. Again it is osteoconductive and bioresorbable, helps in hip and knee joint repair. The next one is Calcium 6, where it is made up of calcium sulphate, osteoconductive and bioresorbable, it has properties, again bone void filler, and it provides strength. These all the ceramic and ceramic derived composites which are used for bone graft substitutes. As I said in the first second session, the biomaterials, where osteoinductive biomaterials, ceramics plays a role. As in the to be straight, ceramic based materials are very much used in bone graft substitutes, because we already know that bone is a organic bone is a composite material which is made up of inorganic hydroxyapatite. Again hydroxyapatite comes under ceramic family, as well as the organic collagen matrix. So, there are many works based on ceramic based materials, and the next one is Norian, where it has been prepared by monocalcium phosphate and tricalcium phosphate and calcium carbonate. It gives very good compressive strength and used in skull bone defect and craniofacial reconstruction, and also it is an injectable paste format. And the third one is tissue replacement, where they have used polymethyl methacrylate, which is a synthetic polymer, and so ceramic based, now it is a synthetic polymer based bone graft material, and good strength, durable and surface osteoconductive. It is used for craniofacial reconstruction. The next one again a ceramic based material, alpha BSM, which is made up of calcium phosphate cement, and it shows good compressive strength. It is used in dental application for bone and cartilage defects. The next is Scorpio OS paste, and which is made up of calcium phosphate and type 1 bovine collagen, natural and synthetic composites polymer, and it provides significantly more calcium and phosphate ions at equilibrium than either of beta TCP or HA hydroxyapatite. It acts as the osteoconductive scaffold for the growth of new bone, osteoconductive scaffold. And the collagraft, which is the mixture of tricalcium phosphate, bovine collagen and hydroxyapatite, again it is bioresorbable and osteoconductive. It can be used for the treatment of long bone fracture and void filling. And the last two one is mainly based on growth factors, which I already told, like BMP2 and BMP7 are FDA approved ones based on OP1 putty, and BMP2 along with collagen matrix, and it is highly enhances vascularization, osteoconductive, and helps in healing of bone fracture. And the next one OP1 putty, which is again BMP7 with collagen matrix, osteoconductive and bioresorbable. It helps in the healing of bone defects. So, there are so many bone graft material approved, and they are still in progress. I have just picked up few bone graft materials mainly based on calcium, and few on polymer and calcium ceramic, and few on natural and synthetic polymer, and some FDA approved bone graft substitute based on growth factor. growth factor strategies. And the cell, since the first one is fresh bone marrow cells, since it is autologous, no need for FDA approval. And the current trends in, so that is all about the commercial available bone grafts and few examples of commercially available products. And now the current trends in bone tissue engineering. So currently people have shown more interest in developing mechanically strong porous scaffolds that can retain proper vascularization on osseointegration. The major pitfall of bone tissue engineering is lack of sufficient and timely vascularization of the scaffold. For example, if you place a scaffold in the construct, see there should be an immediate acceptance of the construct with the host, or the vascularization should happen deep into the construct. It should not stop at the upper layer of the construct. So it happens at many stages, and there are many failures because of this kind of issues. So vascularization is a very important phenomenon or a strategy for an successful ideal bone graft material. So we should know about vascularization. So vascularization is nothing but the formation of new blood vessels. So the greatest amount of new bone formation occurs in the most vascularized area, whereas inadequate vascularization at bone defect sites also associated with decreased bone tissue repair and regeneration. So vascularization has been identified as the major pitfall for the successful bone tissue engineering grafts. So what people have done is, to enhance vascularization, we have strategies proposed to enhance vascularization, thereby to increase the success of the bone graft material. So scientists have proposed several methods to accelerate onset of neovascularization, which is nothing but the formation of new tissue for the survival and integration of bone grafts with os tissue. That includes scaffold design. First, in scaffold design itself, they have changed many fabrication techniques, where they have started doing 3D printing, where we have seen few examples of first generation of second generation scaffold, biomimetic scaffolds, which are nothing but 3D printed scaffolds done by fusion deposition modeling, and inclusion of angiogenic growth factors for the enhancement of endothelial cell. And there are two techniques: in vitro pre-vascularization and in vivo pre-vascularization. In vitro pre-vascularization is nothing but co-culture of endothelial cells and osteogenic cells in vitro, and then we transfer into in vivo and check for the vascularization. In vivo pre-vascularization is nothing but there are two modes where we place the scaffold, in vivo techniques, where we place the scaffold in an vascularized area subcutaneously or intramuscular region. After 2 weeks or after certain time, we harvest that scaffold out and we place that scaffold in the bone defect. But this method requires 2 surgeries. So first we need to place the scaffold to get it vascularized, then we have to remove that scaffold and place it on the defect site. So it requires lot of pain, surgeries, time and everything. And the second method is like where we have vessels which are responsible for jugular veins. For example, jugular veins we place inside this construct and we place in the in vivo, whereas the second method happens to be kind of successful, although but still it is very unclear which method is best for the successful in vivo application, and maybe the combination of all the methods may prove to be more effective for enhancement of vascularization. Then about vascularization, now again about immunomodulatory strategies is becoming increasingly popular for modulating the os foreign response, body response, that is fibrous tissue encapsulation. See, once the construct has been placed, immediate reaction will be inflammatory response, followed by the fibrous tissue encapsulation. So we need to modulate that. So incorporation of immunomodulatory strategies, it is becoming popular to avoid that kind of encapsulation. and that is one of the current trend where we researchers is ongoing in bone tissue engineering. One is mainly on vascularization techniques and scaffold design, and in incorporation of immunomodulatory strategies. Then the main important challenge or a critical challenge is the availability of animal models. Animal models pose another critical challenge to test various bone tissue engineering graft approach preclinically. And preclinically, load bearing large animal models should generally be used to assess graft functionality, and this is one of the major pitfall where we face in order to check for the bone graft properties. So, in future direction for bone tissue engineering to become a widespread clinical reality, it must incorporate the recent technologies that utilizes all the necessary components like scaffolds, cells and growth factors. So, we need to utilize all the new recent technologies in order to develop a best scaffold, ideal scaffold, and also with the cells seeded with the cells, and also for the growth factors for the successful bone repair and regeneration. And also some of the efforts must be made to establish efficient intraoperative cell seeding methods as well to minimize in vitro culture of the bone tissue engineering constructs, which allows bone tissue regeneration, for in vivo studies. So, these are the few future directions where we need to focus mainly on to develop an ideal bone repair and regeneration. So, thank you. So, overall picture where we studied about the bone, its basic functions and its anatomy of the bone, and the modeling and remodeling of the bone, and the tissue engineering strategies, scaffolds, growth factors, cells, and the commercially available products, and the current trends and the future directions. Thank you.